A very good morning and warm welcome to one and all present here. Honorable Speaker of today's session, respected principals, sir, HODs of all the departments, faculties, and my dear friends. I feel privileged to extend my warm welcome to all the participants who have gathered here today for this session on innovations in computer-aided drug design, organized by Institutions Innovation Council, Sri Vidyaniketan College of Pharmacy, in association with Biovia Discovery Studio, Life Sciences Department, Altum Technologies, Bangalore. I would like to extend a warm welcome to our today's speaker, Dr. Sakshi Bharadwaj ma'am, PhD, Application Scientist, Biovia Discovery Studio, Life Sciences Department, Altum Technologies, Bangalore. Your presence makes this event even more special, and I am grateful to have each and every one of you here. Now, I would like to invite our respected principal, sir, to address the gathering with his valuable input. Please, sir.
A very good morning to all of you. It's a pleasure to welcome all of you to this exciting webinar on computer-aided drug design with Biovia Discovery Studio. Today, we gather to explore cutting-edge tools and methods
Sorry for the disturbance. So it's a pleasure to welcome you for this exciting webinar on computer-aided drug design with Biovia Discovery Studio. We gather to explore cutting-edge technologies and methods that are transforming the landscape of the drug discovery and pharmaceutical research. To begin, I would like to express my sincere gratitude to the Desert System and Altum Technology Private Limited Bangalore for organizing this session and for their commitment in advancing the research and education in life sciences. Their dedication in empowering the scientists and the students with state-of-art of the technology is truly commendable. I also want to extend my heartfelt thanks to our management for their unwavering support and encouragement in organizing this event. Their commitment to fostering the culture of learning and innovation has made possible for us to organize such a valuable session. We are privileged to have with us today Dr. Sakshi Bharadwaj, an esteemed application scientist at Biovia Discovery Studio, who will be leading this today's session. Dr. Bharadwaj brings her with the deep insights in computational modeling, molecular simulation, and drug design. Our insights will help us provide understanding how Biovia Discovery Studio can be utilized to accelerate and enhance this drug discovery process. As we know that the traditional process of drug development is complex, time-consuming, and resource-intensive. However, the advancement in computer drug development has opened up the new origin of the efficient and precise drug discovery. Biovia Discovery Studio, a powerful platform for molecular modeling and simulation, offers researchers the tools that needed to predict, visualize, and analyze the interaction between the potential drug molecules and their biological targets. This session will be instrumental in helping us understanding the potentials of computational drug design to develop new treatments faster and more effectively. I encourage all the participants, especially the students and young researchers, to engage actively in this webinar, absorb the knowledge, and ask the questions. This is an unique opportunity to learn from one of the best in the field, and I am confident that this today's session will spark the curiosity, inspire new ideas, and deepen your understanding of computer-aided drug design. Once again, I thank Dr. Bharadwaj for joining us today, and I also thank all the participants for joining as well as your commitment to learning and innovation. That is what drives our institution forward. And we look forward for an enriching and enlightening session. And once again, I welcome all of you. Thank you.
Thank you so much, sir, for your valuable words. Now it's time to introduce our today's speaker, Dr. Sakshi Bharadwaj ma'am. Sakshi ma'am has nearly 12 years of experience in academics, research, and scientific writing. Ma'am also has hands-on experience in QSAR modeling, pharmacophore modeling, homology modeling, SBDD, and LBDD, docking, molecular stimulation, machine learning, artificial intelligence, including R and Python programming, analytical instruments such as UV, IR, NMR, column chromatography, and LCMS. Ma'am has more than 15 publications, index scientific journals, 5 book chapters, nearly 27 scientific paper publications, and 6 scientific blogs. Ma'am also served as a reviewer for many reputed scientific journals like Elsevier, Science Direct, Bentham, Taylor, and Francis. Ma'am was also state rank holder in M Pharmacy and PhD. Ma'am did her B Pharmacy from Kurukshetra University, Kurukshetra, Haryana. Ma'am pursued her M Pharmacy in Pharmaceutical Chemistry from Punjab PTU, Jalandhar. Ma'am also completed her PhD Pharmaceutical Sciences in Pharmaceutical Chemistry from Rajiv Gandhi University of Health Sciences, Bangalore. Ma'am has interest in drug designing, computational chemistry, molecular modeling, machine learning, artificial intelligence, synthetic and green chemistry. As we move forward with our event today, I encourage everyone to actively engage with ma'am to ask questions and listen attentively to his words. Please join me in welcoming ma'am with a great round of applause. Without wasting any more moments, the session is over to you, ma'am.
Thank you so much, ma'am, for such a lovely introduction. I'm thankful to the organizing committee and, of course, to our dear students for being with us today. So, basically, today's topic is computer aided drug designing with Biovia Discovery Studio, which seems to be very easy. And, of course, if you know the basics, okay, there are multiple bioinformatic tools. Okay, many people are using multiple tools. What is the use of those tools? Why we are using those tools? What is the need? What is the significance of using those tools which can leverage our innovation in the field of drug discovery and development, right? So as we know, drug discovery and development has been completely transformed by computational drug designing nowadays with the help of bioinformatic tools. This seems to be very interesting actually. To identify the therapeutic candidates, okay, which could be targeted or which could extend to the formulation development, right? So, how we can use those tools? What is the significance? Again, I will be starting with the initial slides with the very few basics, okay, what is drug designing and how it would be helpful, where the targets, what targets you can identify and how you can focus those targets to get into the drug discovery and development process. What is the significance and how the Bio Discovery Studio can leverage your innovation in the field of R&D when you are looking for the formulation development, right? Followed by a video demonstration. If you feel any questions, please be free to ask me. I am welcoming all the layman questions too, because at a student's level I can understand. But yes, those layman question is the building blocks for you guys, right? So don't hesitate to ask me any questions as I am taking you through all the tools what the Biovia Discovery Studio is having. But of course, as we have some time constraint, I might be little faster, but you can reach me out later at any time to ask your queries too, right? So let me move forward with our slides, okay, without wasting our time. Okay, so let me share first my screen. Um, let me check what are this entire screen. Okay, and share. Please confirm me whether my slides are visible to you or not. Is it visible? Okay,
Yes.
considering it is, yes, considering it is visible to all of you, right? So, as our tagline itself is accelerate scientific innovation, yes. So, we are into accelerating your scientific innovation with the Biovia Discovery Studio tools. Starting with the very brief introduction about my company's profile. Being into Altum, we are a decade-old company where we are basically providing a 3D innovation platform. We are dealing with the engineering and life sciences softwares and hardwares too, where we have the 3D bioprinters, scanners. As a part of life sciences, we have Biovia Discovery Studio, Cosmotherm, Material Studio, Pipeline Pilot, and the ULM, that is the Unified Lab Management. As moving forward with the slides, I'll definitely go through those as well with our different multiple partners, right? So Biovia basically provides a scientific collaborative environment for the advanced biological, chemical, and material experiences. How we are doing that? Socially supporting the next generation scientific innovation, right? In the field of industry, we are addressing scientific driven industries. We are providing the experiences in the different field like biological, chemical, and materials, right? So that is the brand, what brand is actually providing to this 3D environment with the help of this 3D environment. We are only leaders that deliver both virtual and real world, including the laboratory and the in silico part predictions, I would say, which can connect to accelerate the novel therapeutics and materials in the field of R&D and of course the commercialization. So how we are doing? We are taking a problem statement from the real world. We are initiating something to solve that problem statement with the help of virtual field, that means the 3D environment, and again suggesting some kind of a solution to that. We are coming back to the real world. That means these 3D experiences, the 3D platforms, right? So these are the predictions. Anyhow, you have to come back to the real world to implement those 3D predictions, right? So that's what we are dealing with, our doing with our 3D prediction or bioinformatic tools as well, right? Delivering scientific solution experiences in field of biospheres and material spheres, that what we are providing the solutions for science, process, data, and content management as well. This is a slide which just to show you the rich portfolio of Biovia, where we are dealing with the unified lab management. So it is a complete in bioinformatic solution to manage your labs. Okay, comprehensive in terms of comprehensive lab management, informatics, process production operations. We are taking care of quality and regulatory management. As a part of collaborative science, we are dealing with the predictive models, novel therapeutics, engineered materials, and again with the chemicals and formulation. Now, how one can catalyze their research with the help of Bioware solutions. So you can see on the slide, we have 6 major softwares which comes under the Bioware. First is the Discovery Studio, which helps us in the field of drug discovery and development. Cosmotherm is a software which you can use to analyze when you are dealing with multiple solvents or there's mixtures basically. Okay, so you can check the solubility, you can check the partitioning. Even we have the virtual screen module also in the Cosmotherm. Again, in the 3D environment, right? So again, we are dealing with the thermodynamic parameter to make it sure whether whatever we are doing it's correct or having some kind of an accuracy or not. Material Studio, again, when you are dealing with the materials like polymers, co-crystals, batteries, all those kind of things we can do with the help of materials to do in the 3D environment. Simulation is the most important part of Biovia, so you can analyze in real time motion, what we call it as a real time motion check, okay, with our different solutions. Pipeline pilot is basically where you can use a large data set. To aggregate all those large data set is not very easy task, hence we have the pipeline pilot as a solution for all those, when you are handling the different omics data sets. Even the NGS sequencing is a part of pipeline pilot. You can have the image analytics and so on. There are multiple functions of each of the softwares. I'm telling you very briefly. 3D bioprinters, again, whatever the lab work you are doing in the 2D environment, you can replicate those into the 3D environment with our actual solution. That's why we have the 3D bioprinting with us. Unified lab management, again, it has the multiple solution like which can replicate or mimics your daily life works, what you are doing in the lab till you are sending your reports to your manager, and all those kind of issues can be handled well with the unified lab management solutions here. Now coming to what is drug designing, as every must be everyone is knowing that, but still I just want to introduce why we are doing a drug discovery, why we are designing the novel molecule. Okay, so basic funda is finding a new medication, right, based on the knowledge of our biological targets. Now biological targets could be anything, there are multiple biological targets, right? So all those kind of finding medications, what we are using the knowledge of our biological target, that we call it as a drug designing, right? It could be, there are various terms. It could be a drug purposing, drug mining, or so many things, right? In silico, why it is called as, because as we are using the bioinformatic tools, of course with the help of computers, so computers are using some silicon chips, that's why we call it as a in silico. It's a Latin term called the in silicos, right? So that is the concept behind calling it as a in silico. Now there are two ways of doing drug discovery. One is a conventional, that is the chemical lab. Second is in silico drug designing. Now when you are going and doing your effort of doing multiple reactions in your actual lab, that is the conventional lab, you don't, if you have an idea, that is the great thing, right? But usually when the people are going in the commercial lab or any this conventional labs, they are doing the multiple reactions without any directions, hence we are generating the garbage, what we call it as a garbage. Why? Because you don't have any idea whether whatever the reaction you are putting, whether it would be giving you the successful candidate or not, right? To replicate those with the in silico, with the help of bioinformatic tools, we are getting maximum, I usually told it as like 95 to 92 to 95% accuracy. That's why it is called as a prediction, right? So these 3D environment will give you that much of prediction. That means whatever the molecule or whatever the synthesis or whatever the candidates you are planning to get it, you will getting that much accuracy in your results. And again, you are coming back to the wet lab to synthesize only that particular molecule again, because that molecule could be your lead molecule or a drug like molecule. So how the way we are dealing with the in silico and how we are saving our time and cost which we are investing in the different chemicals, right? So that's how, that is the significance of using the in silico drug designing, or that's why we call it as a rational drug designing as well, right? So it's a very generalized PPT I just mentioned just to make it a quick review about it, because first we need to identify a disease condition. This is the cycle which exactly telling us how much time a drug candidate is taking to get into the market, right? Along with all the approvals and accuracy or validation and toxicity parameters work on that, right? So you can ask, we have the multiple drugs molecule in the market, right? So usually I'm taking an example of anti-tubercular drug. We have multiple drugs for that. But still, there are multiple complexity associated with those kind of an drug molecule, even in the terms of drug discovery, right? It should be selective, it should be targeted, it should be potent, it should not be toxic at all, it should be patentable, and of course it should be manufacturable in the market, right? Then only we can have a candidate in our hands, right? So to overcome all those kind of complexity, not even that, we have some kind of problem associated with some drug delivery systems as well, right? So as I was mentioning, the antitubercular drug, we all know that those kind of drugs are having multi-drug resistant problem associated with them, which is now extended to total drug resistance. So to overcome all those kind of problems, we are keep on looking to get one of the hit molecule in terms of novelty, in terms of potency, in terms of bioaffinity, so that we can have less toxicity and more potent molecule as compared to the present ones. That's why we are keep on struggling with to get a one hit molecule. Now, as we are doing the in vivo, we are doing the in vitro as well. Those experiments, in vitro, basically whatever we are doing in labs, and in vivo, when the life living organism is involved into that. So there is a one solution in between these two, that is the in silico, where you can use these bioinformatic tools to predict what could be the good candidate before going into the lab, right? Again, that's what I told. Again, we have to come back to the real world with some kind of a predictivity, that okay, if I am taking this kind of molecules, targeted my macromolecule, this will be showing good affinity towards each other. Hence, we have to synthesize, or we have to take that molecule forward only among all the series of candidates. That's how we are working with the in silico prediction. Where the technology impacting, we just saw the a standard slide of a drug discovery process. I mean, so your candidate coming to the market. Now, where we can impact this in silico technology for this, drug discovery and development process, where we are exactly looking for a drug candidate along with the multiple approvals, and finally when a drug is comes into the market. So the idea started with identify a disease condition. As we all know, we have an example of COVID, right? Epidemic. So again, we need to isolate the protein structure to work on it, right? So, but how we can utilize these in silico predictions or our bioinformatic tools, so that we can optimize or identify those targets, even the ligand molecules, to help us make this process faster. That's why the high throughput screening comes into the picture. You can take large data sets of your molecule, you can screen out your libraries by applying some kind of filters, and you can have top molecules which are the lead molecules which could give you the hit molecule, right? Again, combinatorial libraries is also a part of your this processing, where you can generate multiple ligand molecules with the combinatorial libraries, while providing your reaction format files along with your reagents. Those kind of an tools also we have in Biovia Discovery Studio. Molecular modeling, you can modify the structure along with your, then you can check the multiple activities parameters. And again, you can have, even we have the pharmacophore robust algorithm for our molecular modeling purposes. So we have the multiple ways of doing these kind of processes to leverage the innovation with the help of acceleration, right? That's why we are using the in silico prediction here. So, as this is very obvious, what kind of significance the in silico predictions are having, like, of course, they are saving our time, they are saving our costs, they are enhancing the target and drug candidate identification in a very shorter time span, of course, along with the validation. We can improve the lead optimization with the help of these bioinformatic tools. We can predict there is toxicity and adverse effect prior go into the convention lab or any this assay labs. The most important significance of using these tools is your personalized medication, and when it comes to the biomarker discovery, right? So these tools can give you such personalized medications also when we are dealing with the drug discovery and development process. Again, we have the solution which can enhance the regulatory compliance with the for this process. This interaction or these there are multiple tools I'll show you as we are moving forward. But yes, these tools can support when you can identify the different interaction or various predictions. On the basis of those, these tools can support the mechanistic insights also for your drug discovery and development process. So it's a very generalized CAD workflow I just mentioned here, just to make you guys understand there are multiple ways of doing it. But the main question is where to start, right? So again, you have to start with the already available data sets, right? Or either if you don't have those data sets, if you only having your ligand molecule or the structure of your macromolecule, that is also good enough to go forward. But usually it is suggested because we need to study a lot before going for any kind of a CAD work, right? Then only we can analyze our structures, whether it's a macromolecule or a small molecule structure. It's a part of your virtual screening where you are dealing with the ED, and the TopCat different modules or tools. Even we have some filtering tools also in Biovia Discovery Studio, which is a part of virtual screening of small molecules to get one hit molecule. Even docking could be considered as a virtual screening method to select your top molecules. That could be your lead molecule or hit molecule, right? Okay, now we talked about what is the significance, how it is started and all, but what is the component? So basically there are two components of drug designing when we are dealing with the in silico prediction tools or bioinformatic tools, right? One is search algorithms and second one is scoring function. So, which tool you are using, whether the algorithm is robust enough to do that work, whether it is good enough to validate your result, whether it is good enough to give you the accurate result or not, that is the main building block of your any bioinformatic tool, which will help you to predict the best model. Again, to find out the best conformation in terms of in between the ligand and protein interaction. That's how we are looking for different conformation so that we can select on the basis of those to do the rigid and flexible docking and of course the molecular dynamic simulation. Now, once you have the algorithm, you can do the work, but how to prioritize your hit molecules or the lead molecules out of those data sets, that is the part of your scoring function, where you can ranking the model based on their best fit values. So again, multiple tools are having multiple data sets. So it depends upon which type of tool you are using and how the data set is coming. So that is a separate part. I'll show you as we are moving forward. We can rank the ligands according to the interaction energy, of course. So that's why we are looking for the docking and molecular dynamic simulation, how my molecule is interacting with the targets, right? Whether it is sustaining those interaction while going for the real time in environment with the thermodynamic parameters. So hence we are looking for all those parameters. Hence we are looking for that's why we are applying the force field also to check those kind of parameters. Basically the energy of your molecules when we are taking it to molecular dynamic simulations. Okay, now coming to Biovia Discovery Studio capabilities. I'll take one by one. Okay, so we have five broad modules in Biovia Discovery Studio. First, we have macromolecules, right? So macromolecules means any targeted molecule. It could be your hormones, it could be your enzymes, it could be your RNAs, it could be your DNAs, it could be your anything, right? Whether it depends how big it is. So we can handle any macromolecule here with the help of our robust tools and algorithms. So first is the part homology modeling. So usually we are using the homology modeling when we don't have the 3D structure of our macromolecules. So why we need this 3D structure? Because as we are using the 3D environment, hence we require a 3D structure of your inputs, right? Hence we are looking for those 3D structure. Either you can download it from the PDB data bank or any data bank if it is available there. If not, then you can also generate those 3D structure with the help of homology modeling robust tools, right? Again, we have the protein docking. Okay, so homology modeling, we have the blast, sequence alignment, build, homology model refinement. Even you can define those multiple poses also. That is your models also. That is also possible. Protein docking. Protein docking, yes, protein docking is basically allows researcher, when you are dealing with the two macromolecule. So there are two types: protein docking and protein ligand docking. So protein docking, when you are taking two macromolecules, one macromolecule is bigger and another one is smaller, where you are looking for their interaction, how they are interacting at the interface. So for all those kind of an purpose, we have the ZDoc and RDoc robust algorithm to do those kind of an work for us. Then we have the antibody designing protocols. This process is basically for the vaccine development, and even if you are looking for the antigen and antibody interaction, this module could helpful for all those predictions. Even you can predict the aggregation sites, epitome detections. That is also the part of our antibody designing tool. Protein engineering. Basically, this tools enables to design and optimize protein for the desired functions. You can edit the protein structure as as per the requirement of the different projects. That is also possible to improve its basically for the stability, specificity, binding affinity, or the catalytic activity. So for all those kind of purposes, we are using this protein engineering tool here. Then we have simulation. Okay, I'll come it to later. First, I'll take this small molecule. Small molecule module is having various tools. One is for the virtual screening, where we are using multiple filters to cut down our data sets or mine our data sets to few molecules, so that we can only work with those kind of molecules to get one hit molecule, right? That's what we our ultimate goal is when we come with the virtual screening protocols. QSCR. So this QSCR in Biovia Discovery Studio is very robust with our machine learning and artificial intelligence algorithms. We have various method of doing that. I think I have kept one of the slide to explain all those methods here. Okay, even we have some kind of tools which can that's what I'll just mention. You can give the reaction format along with your reagents here to generate different combinatorial libraries, and again you can analyze those kind of libraries also. ADMET and toxicity prediction also is one of the part of your virtual screening protocol. I'll mention this in the demonstration also. So that's what we are having. Again, these different modules is under each category we have the multiple tools to do that one, right? Simulation. If I'm coming back to the simulation, so here the simulation is a part of quantum and the molecular mechanics combination. Anyhow, we have incorporated that Gaussian dynamics also in Biovia Discovery Studio, which could be very fast. We have some free energy calculation tools also now to check the different energies of your molecule, whether it's a simple complex or your single molecule. So on the basis of those energy calculations also, you can predict the behavior of your molecules. That also we can do and with our simulation modules. Then we have pharmacophore. So pharmacophore, with the help of these specific pharmacophoric features, okay, which is responsible for a molecule to be active or inactive. How to identify those kind of pharmacophoric means those features? For that we have the pharmacophore module here. You can go for the pharmacophore profiling. We have the fragment replacement. You can generate the multiple fragments out of one of the active molecule, and you can take those fragments only on the basis of some scoring function. Here is the Doody score functions we are generating. On the basis of those scores, you can select one of the top fragment and you can regrow that fragment. That is also possible. You can take here the multiple, I think I have mentioned in this particular section, I'll explain there. Okay, whether which type of data set you can take to generate those pharmacophoric features here. And finally, we are looking for reaction, your receptor and ligand interaction. That is the talking. Okay, where we are looking for how it is interacting with my macromolecule, right? So we can handle any type of macromolecule here. Even the membrane molecules also can be handled very easily. Again, the scoring and analyzing those trajectory is, of course, we need to analyze those trajectory in terms of whatever the result we have got. How we are, again, we have to analyze those results to select one of the complex or one of the molecule. However, we also having the X-ray diffraction protocol in Biovia Discovery Studio for your macromolecule structures. So, on the basis of these, we can conclude that Biodiscovery Studio can handle bioinformatics and cheminformatics both in a very good manner, right? Okay, starting with my macromolecule tools, what we are having in macromolecules with us. So, as I was mentioning, macromolecule tools provide rich functionalities that basically allows you to work with the protein sequences. Okay, again we have the separate sequencing protocols. I'll show you. I have kept one of the slide with the unique Biodiscovery Studio feature. There I have mentioned few features which exclusively we have in Biodiscovery Studio. So there also we have one of the protein sequence tool specifically that is the unique feature, and those structures as well as nucleotide sequences and the structures we can handle under this particular category. The main function of those kind of tools, basically either you can download your macromolecule directory from any data bank. We have the URL category for that, or if you have already those molecules, you can pull out there either the nucleic acid or the sequences databases, whatever. So you can pull out those molecules. You can align, as I was telling about the homology modeling, we have the multiple tools to do that as a homology modeling where the modeler is playing the role. So that's what I mentioned that for an software to do work, algorithm is the most significant part. We are using multiple algorithms for multiple tools, which makes our software more robust, okay, while doing these drug discovery and development processes. Create protein model structure using the homology modeling I just mentioned. And of course, you can define those structures also where we are using charm based ab initio approaches. Okay, so you can build and edit the protein, even the nucleic acid structure here. You can prepare the protein structure for other simulation and calculation, and of course, multiple docking experiments also. You can create the model of your macromolecule, or if you have downloaded macromolecule, you can check the different mutations. You can calculate the energies also for basically for the protein stability, specificity, binding affinity, right? So as I was mentioning under the protein engineering protocol, so for all those kind of things, we have the macromolecule tools. Even the different docked protein to protein complexes also we can handle very easily with our macromolecule tools. So I just mentioned what are the tools we are having in Biodiscovery Studio. Let me take a laser pointer here. So you can see we have the macromolecule. So this is the broad category of the module. Under this particular module, we have the multiple tools. I just mentioned few. Okay, so you can align the structures for aligning purpose. We have the separate category for docking proteins. We have the separate category for the general purpose where you can do the editing and all. We have the separate category homology modeling where we are using the modular to do the homology modeling. You can verify those models with our verify protein, that is the profiler 3D. You can handle the antibodies with these multiple tools. Okay, even I just mentioned that we can handle the membrane proteins also. So for that we have a specified tools. Protein designing, even you can edit or build the mutants also. You can calculate the aggregation scores. So all those kind of tools we are having under the macromolecule category. So these are just the picturization. I think I have mentioned many more. Yes. So target analysis, of course, is the major task when we are going for the computer-aided drug designing. Why? Because if you actually understand your macromolecule structure, you can work on it, right? That's why the first step and the crucial step is to understand your macromolecular structure. If you understood how if you gather all those information, it would be very easy for you to understand how where you have to take those macromolecule. That's what I always mention, that the problem is statement. must be very clear when you are going for any kind of a project with the help of these kind of techniques. Why? Because if it is not clear, okay, what you are expecting from those kind of solutions, what kind of results you are actually looking for, what type of results you are looking for, all those kind of question you should understand before going for your these kind of predictions, right? Hence the optimization or identification of your macromolecule is little crucial. If you know those molecules, if you have, these tools will help you to understand or to identify and optimize the molecules, but how and what you are looking into it, that is up to the user only, right? So target analysis, protein and sequence database mining we can handle, modeling, feasibility assessment we can handle, pocket identification of course is a part of our receptor and ligand interaction, where we can assign the binding sites over to your macromolecule. Of course, the mutation analysis with the different type of results, like we are getting some dendrograms, we are getting some multiple, mutation energy with the different, I would say, the tables in terms of each and every protocol here, right? So why we are generating multiple data sets here, along with the statistical data and all, because to prove that whatever we have done it is correctly. It's not just like it we have some kind of an evidence in terms of the statistical data with us to prove our project. So again, the coming to this homology modeling, so you can generate the multiple map views. With the help of these maps you can identify how much is the alignment with of your homologous molecule to your query structure. So all those kind of views we are having. We are generating the beta sheets and alpha helical structure with this color identification, where this blue one is your beta sheets and this one is the alpha helix. So likewise, template identification is the part of homology modeling. You can take the multiple sequence alignment also, a single, but depend upon the identity, how much you are getting. On the basis of those we are selecting sequences to align with each other, right? Even we have the loop refinement tools also with us, so you can define your loops or the side chains also with our robust algorithm, where the looper is playing the role, right? So antibody modeling in the Discovery Studio, basically we are doing, as I just mentioned, that we are looking for the multiple issues when we are dealing with the antibody and anti-designing protocols. Basically, for the mutation and humanization kind of studies, we are using these kind of tools to calculating such kind of like solvent accessibility, electrostatic potential, aggregation hotspots, right? So, as a part of your specificity and your stability of your complex, basically, even we can have this antibody antigen docking studies also with our tools, right? Coming to the small molecules, so small molecules, we can handle each, whatever the format you are having, can you handle each of the format here. But of course, the mole and SJ format is the standard format. But anyhow, you can pull out your molecule in a ligand molecule, basically in any format in the Biovia Discovery Studio. So these are the multiple tools what we are having under small molecules. We have the ADMET, where you can have these TopCat and ADMET descriptors. You can generate different conformations also. Under general purpose, you can prepare your macromolecule. You can do the library designing and analysis. QSCR is we are having the multiple methods of doing QSCR generation. We have the Bison, GFA, MLRA, and PLS. So all those methods you can use. Depend upon what type of data sets you are having. You can calculate or you can analyze different SARs of your structure with the help of these tools under the small molecule category. Right. So what is the purpose of doing these things is because we are actually looking for a hit molecule from the lead molecules, which lead molecules we are getting from the different ligand molecules. When we are having a huge data set of ligand molecules, we are doing some kind of an optimization. We are putting some kind of an filters with the help of different tools, and we are selecting some of the lead molecules. Now those lead molecules will going to lead the series to get the hit molecule. Hence that is the purpose of selecting or doing those kind of an optimization when you are having dealing with the small molecules. Now there are multiple multiple ways of doing it. Pharmacophore is one of that. Virtual screening, docking. Okay, so QSCR, even the kinetic modeling, so top cat analysis, that is also one part of your small molecule drug designing. You can see we have, under top cat category, we have 14 different animal models with the different, you can see here, we have different animal models. Even on the basis of different doses form, you can select one of the model, depends upon what type of work you are having, and you can analyze the toxicity according to that, right? Even we have the male and female category also. So as I was telling, ligand protein, even you can screen out with our toolkit for the development of flexible virtual screening campaign. What will give you the virtual screening? Actually, the virtual screening, that's what, from billions of molecules you are selecting few and then to cut down to one of the hit molecule, right? Ligand preparation. Okay, so before going into the docking or molecular dynamic simulation, once you have selected your few lead molecule or your lead molecule for going for the docking and simulation, we need to prepare our inputs, right? Hence, we have those kind of tools also for preparation of your macromolecule and for preparation of your ligand molecules. Lipinski rule, Weber's rule, SMARTS, molecular properties, pharmacophore, also a part of our small tools here. Okay, which considered as rule of file, basically when we are looking for a drug like molecule in the drug discovery and development process. Protein active site identification is also a part of our receptor ligand interaction protocol, where you can identify or you can generate the binding site of you over to your macromolecule. So yes, we can analyze different non-bonding interactions also with the help of our this receptor ligand interaction protocol. Coming to the QSCR, as I was mentioning, we have the multiple methods of doing QSCR, or to generate different, models on the basis of, basically this quantitative structure activity relationship. That means your structure of your molecules with the multiple descriptors, how it is contributing towards the activity, that what we are analyzing with under this particular category, that is the QSCR. Now, doing that, we have multiple methods depending upon your data sets, whether it is a categorical form or whether it is a regression data sets. On the basis of that, we can select the multiple methods to analyze and to generate your KOCR model, right? So that's what we can map the molecular, I would say, the pairs and activity cliffs here. That's what I have just shown you under the SAR category to analyze your KOCR, right? Basically the activity, what is the responsible for the activity of your molecule. So I just mentioned a few descriptors. You can see these are the tools of Biovia Discovery Studio for creating QSR model. We have multiple descriptors under 1D, 2D, 3D categories. We have some other descriptors also. Even we have the fingerprint algorithms for those kind of descriptors, like we have site eject extended. So again, when you are doing these kind of QSCR modeling, we can have multiple. I'll show you. We have this fingerprint algorithms for doing those kind of projects. Okay, here I just mentioned. So I just briefly mention about multiple methods what we are having. What is the dependent variable? Okay, what is the independent variable, and what remarks we are having for each methods here. So MLRA and PLS is basically the historical methods which are using the continuous data sets, but these methods cannot handle the fingerprints of Biovia Discovery Studio. But we are having these methods. But these methods are over prone to the overfitting, and that's why we are not using these methods, as these are the historical methods and we are having more advanced methods over to them. So we have the Bayesian models, so which is using the binary classification in terms of numerical data sets. It can handle the fingerprint region. Even we have the GFA algorithm with the continuous. So these are, this recursive can be handled the categorical data. GFA for the continuous data set, Bayesian for the binary data set. So all different methods can be used for the different types of data set in Biovia Discovery Studio, along with the different types of fingerprints, right? So Now, diversity and multi-property optimization. Again, we have PCA as a part of small molecules. You can generate different graphs, right, on those component data sets. Cheminformatics, when it comes into the picture, we are looking to mine the data sets on the basis of different analysis. So for those kind of a different analysis, we have PCA, we have ADMET, we have the TopKit, we have the QSCR. So again, some kind of a clustering is a part of our small molecules. Also, we have the different Pareto methods also, where the module is having multiple methods to analyze or filter your molecules. That is also can be done with the help of Biodiscovery Studio tools. Pharmacophore. So pharmacophore is basically, I would say, the first feature to understand how a molecule is behaving. On the basis of those pharmacophoric features, only you can identify your molecule. That is also possible. For example, for anti-tubercular drug, we know the isoniazid, we know the ethambutol, we know the rifampicin, but all the three molecules are having the difference, in a huge difference in their structures. Okay, but still they are behaving as a anti-tubercular drugs. So what is the reason of that? That means some kind of pharmacophoric features are associated with all of them, okay, which are making them as an anti-tubercular against the same category of disease condition. That's what I meant to say, right? So pharmacophoric features you can develop from the active molecule if you don't have a list of your ligand molecules, and you can utilize those pharmacophoric features to screen your libraries with our different algorithms. So here we are having the catalyst algorithm for the pharmacophore development. Under catalyst, we again categorize the hip hop and hypogen algorithms. I'll show you the different slide for that. Okay, so basically what we are trying to do is we are trying to generate different pharmacophoric features from the molecules, whether it is active molecules or the series of your ligand molecules, either on the basis of their biological data, or if you are not having a biological data set, that is without the biological data set molecule, that is also possible to analyze or to generate the pharmacophores out of that molecule, right? Here we are also having the 3D pharmacophore generation, so that is also a part of Biodiscovery Studio. You can see we have multiple tools here. Okay, under pharmacophore module, you can compare the pharmacophores. Ligand profile, again, it's a unique tool of Biodiscovery Studio, where you can generate the multiple pharmacophores, or you can take the multiple ligands against the multiple macromolecules to analyze their polypharmacological activity. You can screen the libraries once you have got your pharmacophoric feature. Whatever the molecule is fitting over to those pharmacophoric feature, that could be your hit molecule out of those features what you have generated with the tools. 3D QSA pharmacophore generation, when you have the biological data set with you, auto pharmacophore generation, and so on. Pharmacophore feature, even you can create the feature from the smart. Feature mapping is a tool when you have two active molecule and you want to map those features molecules to generate the pharmacophore. That is also there. Interaction generation, again, so each this drug designing category is divided into two broad categories. One is a structure based drug designing, other one is the ligand based drug designing. So when it is a structure based drug designing, that means you are dealing with the structure of your macromolecule. Ligand means that you are dealing with the small molecules, right? So to generate or to identify or to generate the pharmacophore within the from the binding site of your macromolecule, that is also possible with the help of interaction generation protocol here. So what we are doing there is we are taking inbound ligand which is already available in the pocket of your macromolecule, and we are generating the pharmacophoric features with the help of that inbound ligand. And you can take those pharmacophoric feature to screen out your libraries to get the molecule similar in terms of activity as your inbound ligand. So that is also possible. You can generate or you can build your own data sets here. As you can see here, we can categorize into two categories, like without biological activity and with biological data sets. Without biological data sets, you can use these two tools, that is the auto pharmacophore generation and common pharmacophore generation. Even the feature mapping is also part of that. With biological data sets, we can have the 3D 3D pharmacophore generation protocols. Okay, so likewise, we are generating multiple data sets with the different category actually, right? Qualitative, quantity. So again, it's based on the quality and quantity of your macromolecules. Okay, ligand based is further divided into qualitative and quantitative, where you can use the common pharmacophore and 3D QSAR protocol. Qualitative is basically using the HIPOP and HIPOP refinement, whereas quantitative is using the hypogen and hypogen refinement algorithms. So that's I just mentioned about the this ligand profiler. You can see we can have the multiple macromolecule with the multiple ligands to analyze what kind of an ligand molecule is interfering with the multiple macromolecule in the pathways or not. If it is so, that means it will create some kind of a toxicity, right? Reaction ligand interaction. Again, we have the multiple methods of doing docking itself. Where I have so okay, let me take this one. So this is the module docking. We have multiple methods of doing docking. Fragment based drug designing also a part of Biovia Discovery Studio. You can analyze or you can have the different scoring functions also under this category. We have the different methods, like we have the libdoc method, which is basically the hotspot. Gold is the third party algorithm. C-Docker is charm based molecular dynamic docking protocol in Biovia Discovery Studio. We have flexible docking also. Now, once you have multiple methods, means you have the docking methods. Now you want to go for the docking protocols. But if you don't have any idea about the binding sites, right? So to identify those binding sites, we have the multiple tools here. So we have the from protein cavities, from PDB's data record, from the current selection, where the site search algorithm and flood filling algorithm will help us to generate the binding site over your micromolecule with the help of these tools. Okay, so here I just mentioned briefly about each method. Okay, where the libdog is using the BFGS algorithm. Okay, on the basis of polar hotspot and the apolar hotspot. Gold is a third party algorithm. C-Docker, that's what we call it as a molecular dynamic docking protocol, as it is working the same way as our molecular dynamic simulation is working on the basis of charm based algorithms. Flexible is using the Cyflex algorithm. Okay, MCC, that is the multiple copy simultaneous search. Again, it's a part of the fragment based drug designing. And also we have the ligand fit, okay, on the basis of Monte Carlo conformation sampling. You can calculate the different histogram, I mean, you can generate different histograms also to check the interaction of your molecule with different amino acids of your binding sites. That is the part of analyzing ligand poses here. So that's how we are generating multiple I would say the graphs, histograms, heat maps or statistical data to understand what type of amino acids and how it is interacting with my ligand molecule and how, right? Non-bonded interaction, I just mentioned it because under table also we are getting multiple information, what type of amino acids and how which atom it is connected, what is the number of that particular atom where it is existing, all those kind of an information we are getting under table. I think I have mentioned in next slide. So we also getting some unfavorable interaction. You can understand that also with our 2D interaction tool. Analyzing results. So likewise, I just briefly mentioned here, we are getting this type of information along with the different distance, category, and type of the bond with respect to each complex or different poses. Coming to the protein-protein docking, we have robust algorithm called the Z-dock to do the protein-protein docking here. Even you can define those docking poses with RDoG algorithm. That is also possible. Under this category, you can handle different macromolecule no matter what. Right. Coming to the simulation. So simulation is basically when we are mimicking real-time environment or we are relying on the realistic model. Okay. In real-time motion of your each atom. So basically here we are dealing with the atoms. Hence, we are looking for those real-time motion environment check with respect to the complexes or a single molecule also, that is also possible. Here we are using charm force field. Basically, this is from the lab of chemistry at Harvard Molecular Mechanics, from the Professor Martin Karper's lab at Harvard University. So that is what one of the collaborator of Biovia Discovery Studio for the charm force field, right? So under simulation, we have these categories of our tools. Under analysis, dynamics. So you can see we have the NAM dynamics here. Anyhow, we have incorporated Gaussian also under this category. We have electrostatic potential, that is again a unique feature. Energy minimization, different calculations also you can do. We have the multiple force field here. Free energy calculations also a part of the Biovia Discovery Studio under simulation category. Okay, so you can analyze different, you can generate different solvation also. You can provide the, or you can generate the harmonic reconstant or to a very specified region here, that is also possible, right? So thermodynamic analysis is basically molecular dynamic part, where we are looking for the quantum mechanics and molecular mechanics with the different FEP and DFT calculations. Charm is the part of simulation, hence charm is taking care of all those categories, or it is incorporated with each and every tool here, basically I would say. Explicit membrane based molecular dynamic simulation is yes, you can do that with the different membrane proteins also, that is also possible, where you are dealing with the bilayer of your lipids or any kind of solvents here with the different counter ions. So quickly let you know about the process of molecular dynamic. We need to give the inputs to the molecular dynamic simulation. We need to apply the force field. So basically, why the force field? Because force field is used to describe the forces in between your atom in simple terms. Okay, whether in terms of in energy, of course the functional form of these in terms of energies, right? Different energies which are incorporated with each atom, how they are bonded to each other. So all those kind of energies we can calculate with the help of these force fields, right? Then we need to solve it our system. Under solvation, we are solvating the system and we are adding the counter ions. Then we have the 5 steps of doing this simulation: minimization, heating, equilibration, and production. And finally we are analyzing our RMSD, RMSF, and radius of gyration. So these type of results you can get from our at the end of your trajectory analysis, right? So you can generate these type of plots also. So automatic protein preparation. So that's what we are having. You can generate different. That's what the protein preparation also will take care of all those kind of parameter. If the missing loops are there, protonation define the pH. So all will take care by the preparation tools. Going for the simulation, we have the NAM dynamic simulations also under dynamic category, where you can analyze the different categories. Again, so dynamics NAMD is basically in the GPU mode, means you can have that in the GPU mode also, so that would be little faster. So I just briefly mentioned what is the machine learning algorithm. So Biodiscovery Studio is using is just a brief introduction about our each and every algorithm with respect to the different modules. Okay, so these are the unique feature of Biovia Discovery Studio, where I just mentioned ligand profiler. Even the MSLD comes under the simulation module that calculate the binding affinity with the different combinatorial libraries. TopCat for toxicity analysis, electrostatics of biologics, which calculates the special distribution of your different electrostatic potential on the basis of charm a module. Street molecular dynamic simulation, which is also using the charm force field. Okay, and de novo type of drug designing, and of course we are having the biophysical properties, right? So I just briefly mention about the applications in each category, like in the purpose of drug designing, biotherapeutics, molecular dynamic simulation with the which can handle the different macromolecules, molecular docking, protein sequence analysis, even the molecular modeling. So what areas we are covering with the Biovia Discovery Studio? I just mentioned a list. Okay, so what is new in our this upcoming year? We have the Gaussian molecular dynamic simulations. We already means we introduced the PAN also, which can identify the mimicking molecule which are mimicking the behave like a drug like molecule, but they are not at the end. So all those kind of tools also we are incorporated. So this is what we are our enhancement. I'll just take you quickly for demonstration. Okay, so why we are providing the collaborative platform, we are providing end-to-end solution, and we are providing the solution no code, no pain wala statement, because there are no use of any code. You are not using any code to do any kind of work. That's why it's an easy user interface simulation. This is a simulation platform, and hence you can accelerate your research with the innovation. I'll show you while going for the demonstration how fast our tools will work. What services we are providing? We are solution consultancy, we are providing trainings, customer support, and scientific and contract research also. So these are just briefly a slide about our customers, our clients in each field. Okay. So please keep your questions. Let me directly take you to the demonstration. After that, we'll take care of each and every questions here. Okay, let me share my screen again. I just have one video to show this demonstration. Okay. Okay, so this is the Biovia Discovery Studio interface. We have multiple modules here. Okay, we have tools here, the files category. Okay, so I just took a one of the series of ligand molecules. We have 3,000 of molecule in this. I just quickly show you how we can go for the ADMET prediction of these molecules. Under small molecule category, under ADMET, we have ADMET descriptor to analyze the ADMET of your molecules. So basically, we are looking for the absorption, distribution, metabolism, and excretion, and of course the toxicity with the different parameters of our ADMET. Once you select these two parameters, you can run your protocol, and you can see you will get the result in very few seconds actually. For these 3,000 of molecule, okay, it is done. So ADMET, it's completed. While clicking on this, you will get the results. You can check your results with this view results option. It will ask you about the ADMET plot. Yes, we exactly see that plot. So with this plot, I am not able to understand what type of molecule is passing those parameter or not. But however, you can identify the lesions to see what type of OPS with the different colors what we are having. So these different colors is specified for ADMET, A-log, absorption, and BBB at a different percentage. To filter these molecule, you can go click. Means you can click here on the molecule with the right click, and we have one option called filter here to filter these parameters. We have fixed some kind of levels. For example, for solubility level, we have fixed this level to. We need to keep this as equals to level 3. Okay, then we need to keep the next parameter, that is the BBB level, equals to, it depends, if you are looking for CNS then it would be 0, otherwise 1 or 2 you can keep. Then we have CYP prediction equals to false, right? And hepatotoxicity prediction equals to again false. And we have absorption level that could be equals to 0 for the good absorption, and for PBB, that is the plasma bindings prediction, false. Now you can see we have got a very few molecules out of those 3,000 of molecules after keeping these filters. Now you can take these molecule for topcat prediction, that is the toxicity prediction, right? We have two here, extensive and topcat are working in the same way, but in extensible you can add your you can extend your data sets. Okay, you need to give the molecule here. Models, you can select multiple models, okay, while clicking on those three dots. But for a timing, I just showed with the one model here. Similarity check, it will take three reference molecule in the similarity check. Okay, once you have set this, you can run the protocol. and you will get the results. It will take little time to run these because it is generating the PDF results. Once it is done, it will ask you for to show you the result in the PDF format. You need to click on OK. There we have the multiple detailed report has come here on the basis of different modules, male and female for ball. So you can see the structure with the general information, prediction, it has mentioned non-predicted with the Bison score and other like top features for the negative contribution, even we are getting for the positive contribution also with the three structure of similarity check, right? So likewise, you can have this Topcat and ADMAT prediction. Let me take you quickly to our other protocol, that is the so this is just for the antibody designing. I just want you to see all the different modules here. Further, we will be going for different tools. Let me take one of the FASTA sequence here to do the homology modeling. So once you have this FASTA sequence, you can have the multiple tools under the macromolecule category. First, we need to run the blast so that we can identify the multiple homologous molecules here. We have the multiple input data sets here, but we are taking the non-reagents 95% data sets to analyze to get those hits. Once you have got those hits, you can take those hits depending upon the identity. If you are having one in the good identity, you can take one of the molecule, but if not, then you can take the five of the molecule to build your the structure, right? So likewise we are doing. Once it is completed, okay, you can see we have multiple blast here. So you can see the identity, sequence length, alignment, e values, all the data sets is coming in the scale in the table form. Or even while clicking, you can also open the results. You can view the results, okay, and you can select the multiple molecules in the map view. As I shown you that we are getting the multiple maps with the alignment, again with the different color identification, so you can identify while keeping your cursor on each of the color, you will get the information about the molecules. So likewise, you can see the yellow color is maximum aligning with my query structure. So in that way, you can select the macromolecule for the generation of your building of your homolog means your homologous model. Now I need to select the five molecule. I'm going to load this structure with the alignment for the alignment in my graphical window. So this is basically the molecular window of Biovia Discovery Studio. As the identity is very less, hence I have taken the five homologous molecule to align with my query structure so that we can make sure the structure what we are going to build is with the great accuracy. Now you can see we have the templates also, and we have our molecules on the graphical window here. Now, what next is once we have done the blast, we have done the alignment. Now we are going for the okay. So this is basically for the identification of your beta helix or alpha sheets. We are going to align the sequences to the templates with all of those structures here. So for that, we are using the align sequences to templates tool. Once you have set all these parameters, you can run the protocol for the alignment of your sequences with the different templates here with respect to our query structure. Okay, so let it run. Okay, let me make it a little faster. Okay, so once it is done, you can have the results with you. Next step is to build the homology model. Basically, you need to give the input as a sequence, right? Templates. Model, I'm keeping very low, and optimization level is low because for the tutorial, we are not looking for the great accuracy. Otherwise, you can keep it as a high rate, right? And you can run the protocol, right? Once you have done it, you will get the models. I'll show you how the results look like. Okay, so here you can see we have got the models. Just a minute. So these are the different models what we have got after that particular model. Even run that building model. Even you can analyze the different RMSD values from the structure and RMSD with respect to, if you are having reference molecule, that is also you can pull here and you can identify the RMSD values with respect to each model, and then on the basis of those you can analyze your model structure and you can take your model structure further, right? So I just make it little faster. Okay, so as it is requested for the few modules, that's what I have shown here. So basically now we are going to see how the protein modeling can be done with the help of our ZDoc protocol. So ZDoc protocol, basically we need to take two macromolecule structure. We need to give one bigger molecule as a macromolecule and small one as a ligand molecule. Angular size again 6, giving you the more molecules, means angular size again it will take a very small angular size for each of the poses. This platform, Z rank also is a part of this, but for a timing, we are keeping it as a false. We are generating different cluster here with the multiple parameters. If you are not have know about these parameters, here one help section is coming for those as a beginner. So from there you can read out. Once you set these parameters, you can run the protocol and you will get the result like this. Okay, with the different poses, right? So these are the poses which you can see here. I'll show you in the hierarchy view. So we are getting different clusters. We are getting different docked poses. Okay. On the basis of this color identification, you can identify. So these poses are the good ones with the highest z dog score, because we are getting this rank, density, cluster size, and z dog score also. And as the color is going down, the z dog score is also getting down. You can lock the visibility for the top pose, okay, and you can check with these arrows while keep on clicking on these arrows for the next poses here, right? I just make it fast. Once you have selected the pose, okay, on the basis of your cluster and your zdog score, now I need to refine the top z score poses with the by selecting your top poses here, and you will get the refined poses also. So that is also possible. Okay, so now we have got this uh, top pose, okay, that is the 15. And anyhow, with these poses also you can calculate the RMST with the reference molecule. That is also possible. Let me make it a little faster. Okay, let me show you the result also. So here, after this calculation, you are getting the different energies values in terms of van-der-Waals energy, electrostatic energies, solvation energy, refined octoses energy. So all those kind of an energy is a part of your statistical data here, right? Different energy, you can take a reference molecule to analyze the RMSD value of your poses. Okay, so that is also possible. I'll just take a quick move. Okay. Now coming back to the receptor ligand interaction here. Okay. So I'll just take one of the macromolecule from the URL dataset from the file option. You can get this option. You can directly feed the ID of your macromolecule to open your macromolecule. Okay, once it is opened, we need to analyze its report to check what type of molecule it is, ligand is there, how many chains are there, whether the active sites are present or not, if yes, then whether the water molecules are available or not for at the binding site. Those all kind of an information we are getting from this report. If the water molecule is there, then we need to keep those water molecules. Okay, and once you have got this information, that means we need to keep whatever the sites, for example, this A semicolon some amino acids, I can see some B semicolon some amino acid. That means both the chains are participating in this active site one. Hence, I need to keep both the chains here. So these two always the chains of your macromolecule. This is your ligand molecule, and these two are the water molecule. We need to select some of the water molecules if those are present at binding site, right? So once you have selected the water molecule, simple by selecting and deleting. So now we have selected those. Now the next step is to prepare your macromolecule before going for the docking. So with the help of our prepared protein structure, you can have this prepared structure with us. Okay, where again we have in the hierarchy view water molecule, its side chain and ligand molecule. With the help of the ligand molecule, now we are going to analyze the binding site over this macromolecule. As I already selected, that means you can have the selection option also. I'll show you where those options are to identify the binding site of your ligand molecule. So, if, for example, some of the amino acids are present on the binding site, so you can select those amino acids and you can click on the current selection option also to generate the binding site. That is also possible. But as we know, this PDB site record some information about its active site, so you can take the second option for generation of binding site. But if you don't have any idea about the receptor cavities, then for that we have the first option which will work to identify on your binding sites, right? So with the help of these tools you can identify the binding site. I just taken a already prepared molecule what we have in our database. If you wants to prepare this small molecule, then under small molecule category, we have an option called the prepare ligand. You need to give your library to it, and you need to let it be default set for all the parameters, and you can run the protocol to prepare your ligand molecules, right? So yes, I've selected only few for the C Docker purpose, because C Docker we consider the most accurate because it works with the charm force field act as a molecular dynamic protocol. I just want to show you how we are getting the different results with the C Docker. Okay, once it is done, okay, I missed that part. Okay. So once you clicked on the result file, you can see the interaction here. You need to click on this receptor recent interaction, ligand interaction, after defining this ligand molecule to show on the graphical window. Okay. Under table, we are getting the C Docker energy and minus C Docker energy and minus C Docker interaction energy. On the basis of those interaction energies, we are selecting your molecules. Okay. You can take these pictures and you can have it. Now next is I am taking the simulation. So just a quick intro about the simulation, okay? Because I that's why I have only taken a very small molecule here. So for example, this is my macromolecule. Either you can pull out the complex here, because I just have taken the small molecule. Okay, I just want to change its representation to show you the importance of the force field, how it is working when we are applying the force field. Before applying the force field, and I will check the atoms what this molecule is having. Okay, before that, the structure is looking like this. Once we have applied the force field, okay, we have now we have 436 molecule. Before it was like 200 something molecule in our structure. So this one is the before force field applied, and this one is the after force field applied, right? Once you have applied the force field, next step is the solvation. We need to give our apply typed molecule with the force field, the different models. Cell shape is there, minimum distance of your boundary to your macromolecule. The 7 is for the bigger molecule and 3 is for the smaller molecule. Once you have set this parameter, you can run the protocol. However, we are also adding some kind of a counter ion here for the solvation purpose. Okay, once it is solvated, it will look like this. Okay, next is standard dynamic protocol for the dynamics to run the dynamics. Okay, you need to give your typed molecule after the solvation. We have two robust steps of minimization here. One is the steepen descent, and the other one is we have the conjugate gradient. You need to set all those parameters with the heating and equilibration, and finally we are getting frames in the production. Even you can apply those implicit solvated model here also. So I'm not going into much detail in a 1s stress. For a time being, I just taken give you the quick introduction about the different modules of Biovia Discovery Studio, how we are dealing and how we are taking our molecules for different in silico prediction. If you want to take this results and you want to extend your dynamics with the NAMD, that is also possible. You can take the restart file what our protocols are generating. You can take those restart file and you can extend your dynamics at any stage. That is also possible here. Okay, so that's what I am having for the demonstration. So if you have any questions, you can ask me. I know I moved little faster, but just I want to give you the brief idea about each and every module. So sorry for that.
Thank you so much, ma'am, for accepting our request. That was really a wonderful and amazing session. I hope everyone has enjoyed it and we do learn many new things and perhaps gain knowledge also. Now the session is open for queries. Anyone?
Good afternoon, ma'am. This is Sitesh studying B.Com fourth year. Hello.
Hello. Yeah. Yes.
Yeah, I have one question, ma'am.
Yes.
Can you say what are the some applications of document?
Uh, can you repeat it? I am not able to understand.
What are the some applications of document?
Docking.
Yes, ma'am.
Yeah, okay. So see, docking, why we are doing actually, we are looking to check the different interaction in between a small molecule and a macromolecule. Okay. At that time, we are doing the docking. So docking means we are taking small molecule within the binding site of your molecule. When it is going and docking to the binding site, how it is behaving, what is the interaction, what type of interaction it is showing to on the basis of those interaction we are analyzing what type of how much it is I would say how much it is having the binding towards that macromolecule in terms of interactions. Again, it is the part of the bio affinity of your small molecule towards the targeted. So that is the purpose of going for the different types of docking methods, analyzing different docking poses with the different conformation which we are generating with our multiple tools, or even with the different poses what we are getting with our different methods. So yes.
Thank you, ma'am.
Ma'am, good afternoon, ma'am. I am having one question. How to perform multiple ligands simultaneously docking?
Yes, you can see. I just show you a series of my ligand molecules, right? So those I have selected in one of the parameter I have given as my ligand molecule for visible only, because I have taken only one molecule for the docking purpose. But there also we are getting the option, if you are selecting all, so you can use you can take all the ligand molecules simultaneously for your docking purpose. That is also possible. Even you can run multiple protocols simultaneously with the help of Biovia Discovery Studio. Yes.
Thank you, ma'am.
Good morning, ma'am.
Yeah, tell me. Yes, good morning.
Ma'am, do you have any option for screening the molecule based on Verber's rule?
On the basis of what? Can you be
Verber's
little louder?
Verber's rule.
So you are asking whether you can select on the basis of verbose rules? Yes, we do. We can do that. Yes, we have multiple options in while going for the filtering of your ligand molecules, where we have kept the Lipinski rule along with the Verbose rule. So again, Verbose rule is an add-on to the Lipinski rule, where we you can check the this polar surface area also, where we have fixed it to 140 angstrom. And of course, you can filter out. Again, the Verber's rule will work with the rotatable bonds, which the Lipinski rule is not doing that, right? So yes, you can filter out on the basis of Verber's rules. You're not audible. It's on mute actually. Yes.
Good morning, ma'am. Thank you for the super session. So my doubt was, as a student, it is necessary to learn any course or program to understand the method, reasons, or algorithms. If necessary, can you suggest any course or program, ma'am?
There are multiple courses actually. To if you want to learn the Discovery Studio and development process, yes, there are multiple tools depending upon what type of tools you can avail. So you can adopt any one of them. Right.
Can you name it, ma'am? Any of course, any
So
one?
there are many actually drug discovery के लिए you can have the multiple courses for molecular dynamic simulation. You have the multiple courses now. The machine learning algorithm, which is a part of Biovia Discovery Studio, separately you are getting those kind of courses also. So yes.
Okay, thank you, ma'am.
Ma'am, good afternoon, ma'am. My question was, are there any molecular docking free software for protein and ligands, ma'am?
So free software span. Uh, there are multiple actually. So there are many tools which the people are using and which is online available freely for the user actually. Yes.
Okay, ma'am, thank you. Now I'd like to call upon Dr. Vema Kiran sir to propose vote of thanks.
Good afternoon, ma'am.
Yeah, documents.
Good afternoon to esteemed guests, colleagues, friends, and my dear students, and all others who grace this occasion with their presence. I, Dr. Vemakiran, stand before you today filled with immense gratitude to thank each and every one of you for making this event a resounding success. Your presence, your participation, and your invaluable contributions have truly elevated this gathering to extraordinary heights. First and foremost, I would like to express my heartfelt appreciation to our distinguished speaker for today, Dr. Sakshi Bharadwaj, for honoring us with her presence and delivering such an informative and insightful address. Madam, your words have not only enlightened our students but also motivated us to strive for greater heights. I would like to extend my sincere thanks to Honorable Chancellor Dr. Mohan Babu Garu, Pro Chancellor Manchu Vishnu Garu, Honorable Vice Chancellor Dr. Nagaraj Ramarao Garu, and Dr. Mallikarjun VP, Dean School of Pharmaceutical Sciences, Mohan Babu University, for encouraging in organizing the event. I would like to thank Organizing Committee Dr. Prashanti and her team, whose tireless efforts and meticulous planning have made this event possible. Your dedication and hard work have been instrumental in ensuring the smooth execution of every detail. I would like to thank the technician team of this auditorium for your generous support. Your commitment to our cause has enabled us to create this memorable experience. Last but not least, I would like to thank all of you, my dear students, for your enthusiastic participation and active engagement. Your presence has created a vibrant, positive atmosphere that has made this event truly special. Thank you once again, everyone. Let us continue to work together to create a brighter future. Thank you.
Thank you, sir.
And I request you to answer the queries that were posted in the chat box.
Sita? No, without docking, you cannot calculate the docking score for sure. Option for screening the molecule. Yes, we have. Yes, that I already answered. Do you have the Ramachandran plot? Yes, we have the Ramachandran plot under our tool category, under the chart section. We have the Ramachandran plot where you can analyze or validate the structure of your macromolecule. Yes. What is the optimum time to do molecular? So, sir, there is no optimum time. Actually, it's depend upon the structure of your macromolecule, how many amino acids are there. So, it depends upon the atoms. Again, why? Because we are analyzing the molecule under the molecular dynamics simulation for real-time environment check with based on each and every atom with that thermodynamic parameter. So there is no time limit actually means no optimum time actually. It depend upon the multiple or it depend upon the macromolecule. Again, the number of atoms. Yes. 2D diagram. Yes, definitely. Why not? We have the Biovia draw also. So for your 2D diagram, you can use the Biovia draw that is for free. Yes. Prediction of EDMT is done in normal biological condition. Is it possible to get the prediction for disease model like the different types of cancer? So yes, basically ADMET we are looking for small molecules. So if yes, that could be your answer. Yes, we can do that. Thank you, sir. Any more questions? Thank you. Thank you a lot. Do we have any other questions?
No more questions.
Okay, thank you so much for patiently listening me. Hope I did not bore you guys. So yes, these are the techniques. I have to concise in a very shorter time. That's why maybe I was little faster. So please excuse me for that. But if you have any queries related to any tools, you can reach me out anytime. And for getting more updates on the Biovia Discovery Studio, please stay tuned with us. You can like us, subscribe us to our social media profiles, and you can follow us for getting the new updates. That is also that. Yeah.
Thank you so much.
Thank you. Thank you.
Thank you, ma'am. Thank you so much on behalf of India Regulator College of Pharmacy to Biovia Discovery Studio. One small query, ma'am. Actually, whatever the topic studies we have discussed here regarding amino acids and proteins and pharmacophores, are there any mm, softwares related to polymers concentration optimization in the drug development?
Is there any, ma'am? What?
Polymers,
Um,
polymers.
yes, ma'am. We can handle those type of queries also. But once I will get your problem statement, that would be more clear to me. Yes. Yeah.
Actually, we are using wide varieties of polymers. Wide varieties of polymers.
Okay. Okay.
Like optimization of polymer concentrations for drug delivery.
Okay.
Do
So
you
with
have any softwares?
can you repeat it, ma'am?
Do you have any softwares, ma'am? Software.
Yes, ma'am. For that's what I think initially I mentioned about the material studio. So it depend upon your project. Polymers also we can handle with the help of material studios. So that is also there. So that's what I told. If you can elaborate about your, I would be giving you the solution on that. Yes.
Okay, thank you. Thank you.
Thank